Welcome, everyone. Today, we're going to explore the fascinating world of biological classification. This is the first slide, so I want to warmly welcome you all and thank you for being here. The title of our presentation is Biological Classification, and there's a subtitle that emphasizes its importance in understanding living organisms. Over the course of this presentation, we'll cover how scientists categorize and organize the vast diversity of life on Earth. We'll look at the main principles, the different levels of classification, and why it matters. Let's get started. Let's take a look at this worksheet, which provides an overview of the different kingdoms and types of microorganisms we've been discussing. It covers a broad range of topics, starting with reproduction in fungi, which is quite fascinating because fungi have unique reproductive strategies compared to other organisms. Moving on, we see references to eubacteria, also known as true bacteria, which are the more common bacteria we encounter daily. Then there's viruses, which aren't classified as living organisms but play a crucial role in biology and medicine. The worksheet also highlights the kingdom fungi, emphasizing their distinct characteristics. We also see archaea, which are similar to bacteria but have unique features that set them apart, often thriving in extreme environments. Further, the slide mentions the kingdom Animalia, which includes all animals, and the kingdom Protista, a diverse group that doesn't fit neatly into other categories. Then, we have the kingdom Plantae, covering all plants, and Monera, an older term that broadly referred to bacteria before the reclassification into eubacteria and archaea. Lastly, the worksheet mentions Mycoplasma, a genus of bacteria that are notably small and lack cell walls, making them quite interesting. Overall, this slide serves as a roadmap for understanding the diversity of life forms, from microorganisms to complex multicellular organisms, and how they are classified into different kingdoms based on their characteristics. It's key to see how each group fits into the bigger picture of biological diversity and evolution. Let's take a look at how the classification of organisms has evolved over time, focusing on the different kingdoms and systems used. Starting with the two-kingdom system, proposed by Linnaeus in 1735, which divided life into Plantae and Animalia. Moving forward, in 1866, Haeckel introduced the three-kingdom system, adding Protista as a separate kingdom. Then, in 1956, Copeland expanded this further into a four-kingdom system, including Monera alongside Plantae, Animalia, and fungi. By 1969, R.H. Whittaker developed the five-kingdom system, which recognized Monera, Protista, fungi, Plantae, and Animalia, providing a more detailed view of biological diversity. Finally, in 1990, Carl Woese revolutionized classification again by introducing the three-domain system, based on genetic analysis. This system includes archaea, bacteria, and eukarya, which now serve as the broadest categories. Notice how each of these systems reflects our growing understanding of the relationships among organisms, especially as new scientific techniques like molecular studies come into play. This progression highlights the dynamic nature of taxonomy and how it mirrors advances in biology itself. Bacteria are the only members of this kingdom, which makes it quite unique in the biological world. They are categorized into four groups based on their shape, so you can think of these groups as different physical forms that bacteria take. These shapes include spherical, rod-shaped, spiral, and a few others, and each shape has its own characteristics and ways of thriving. Additionally, bacteria can be either autotrophic or heterotrophic. Autotrophic bacteria can make their own food, much like plants do, through processes like photosynthesis or chemosynthesis. On the other hand, heterotrophic bacteria rely on consuming other organisms or organic matter for energy and nutrients. Understanding these distinctions is really important because it helps us grasp how bacteria interact with their environment, how they survive, and their roles in various ecosystems. This classification under the kingdom Monera highlights their fundamental biological differences and their significance in the broader picture of life on Earth. Now, here we see some fascinating microorganisms that play vital roles in different environments. First, let's talk about methanogens. These are bacteria that are found in the guts of several ruminant animals, like cows and sheep. They are responsible for producing methane, also known as biogas, from the dung of these animals. This process is an important part of the natural carbon cycle and has implications for both agriculture and energy production. Moving on, we have thermoacidophils. These bacteria thrive in extreme conditions, specifically in hot springs and highly acidic areas. They are remarkable because they can survive and function where most other organisms would perish, showcasing the incredible diversity of microbial life. Finally, there are halophiles, which live in highly saline environments, like salt lakes or salt flats. These microorganisms have adapted to survive in conditions with very high salt concentrations, and they often give these environments their characteristic pink or red coloration. All of these groups belong to the domain Archaea, which is a fascinating part of the microbial world, showcasing how life can adapt to even the most extreme environments. Understanding these microbes helps us appreciate the diversity and resilience of life on Earth, and it ties into our broader theme of microbial roles in both natural ecosystems and human-related processes. Now, let's take a closer look at the role of bacteria, particularly the majority that are important decomposers. These bacteria are incredibly helpful in various processes like making curd from milk, producing antibiotics, and fixing nitrogen in the roots of leguminous plants. Their ability to decompose organic matter is vital for maintaining ecological balance and soil fertility. Moving on, we have heterotrophic bacteria, which rely on organic compounds for their energy and carbon. These bacteria play a crucial role in breaking down complex organic materials, recycling nutrients back into the environment. Next, let's discuss chemosynthetic autotrophic bacteria. Unlike photosynthetic organisms, these bacteria oxidize inorganic substances such as nitrates, nitrites, and ammonia. They utilize the energy released from this oxidation process to produce ATP, which fuels their metabolic activities. These bacteria are essential in nutrient cycling, especially in recycling vital elements like nitrogen, phosphorus, iron, and sulfur in ecosystems. Now, turning to cyanobacteria, also known as blue-green algae. These are fascinating because they contain chlorophyll, similar to green plants, enabling them to carry out photosynthesis. Cyanobacteria can be unicellular, colonial, or filamentous, and they are found in both marine and terrestrial environments. Usually, their colonies are surrounded by a gelatinous sheath, which provides protection and structural support. Some species, like nostoc and anabina, can fix atmospheric nitrogen in specialized cells called heterocysts, contributing significantly to soil fertility. Finally, let's touch on photosynthetic autotrophs. These organisms are characterized by having a rigid cell wall, which provides structural support. If they are motile, they move using flagella. They harness sunlight to produce their own food, playing a fundamental role in the energy flow of ecosystems. And now, a quick note on eubacteria, or true bacteria. These are the most common bacteria we encounter daily. They have a simple cell structure, but are incredibly versatile and vital for many biological processes. They are a key part of the microbial world and are essential for life on Earth. Let's take a moment to look at some common bacterial diseases across different domains: plants, animals, and humans. Bacteria can cause quite a range of health problems, but they also have beneficial roles. For example, in plants, one common bacterial disease is fire blight, which affects fruit trees like apples and pears. Moving on to animals, an example would be brucellosis, which impacts livestock and can cause reproductive issues. In humans, tuberculosis is a well-known bacterial disease that primarily affects the lungs. Now, while these bacteria can cause disease, it's important to remember that bacteria also have beneficial roles. For instance, certain bacteria in the human gut help us digest food and synthesize vitamins. So bacteria are not just pathogens, they are integral to many biological processes. This highlights the importance of understanding both the harmful and beneficial aspects of bacteria in our study. These organisms are quite unique because they completely lack a cell wall. This makes them some of the smallest living cells we know of. Interestingly, they can survive without oxygen, which is quite different from many other bacteria. Many mycoplasma are actually pathogenic, meaning they can cause diseases in both animals and plants. This highlights their importance in the broader context of microbiology and health. Understanding these organisms helps us appreciate the diversity and adaptability of microbial life. Today, we're going to talk about reproduction in bacteria. Bacteria primarily reproduce through a process called binary fission, which is a form of asexual reproduction. In this process, a single bacterial cell, or parent cell, duplicates its DNA and then divides into two identical daughter cells. This method allows bacteria to multiply rapidly under favorable conditions. It's fascinating because, despite being simple organisms, bacteria can increase their population exponentially in a very short time. This rapid reproduction plays a key role in their survival and adaptability, especially in changing environments. Understanding bacterial reproduction helps us grasp how infections spread and how bacteria can develop resistance to antibiotics. So, in essence, the process of binary fission is central to how bacteria thrive and proliferate across various environments. Now, let's take a closer look at how single-celled eukaryotes reproduce. They have the interesting ability to reproduce both asexually and sexually, which gives them a lot of flexibility in different environments. This dual method of reproduction is quite important for their survival and adaptation. Moving on, we are in the section about the kingdom Protista. This kingdom is incredibly diverse, including organisms that don't quite fit into other categories. Understanding their reproductive strategies helps us appreciate how adaptable and resilient these organisms can be. It also ties into the broader theme of how different life forms reproduce and evolve, even at the single-cell level. Diatoms are the primary producers in the ocean, meaning they form the base of the food chain and produce a significant portion of the world's oxygen. In diatoms, you'll notice that their cell walls are made up of two thin, overlapping shells that fit together like a soap box. These shells are embedded with silica, which makes them incredibly durable and indestructible. That's why diatoms can accumulate in large amounts over time, forming deposits known as diatomaceous earth. These organisms are found in both freshwater and marine environments, highlighting their ecological versatility. Because they are microscopic and lightweight, they float passively with water currents, essentially drifting along as part of the plankton community. An interesting use of diatoms is in polishing and filtration. The gritty silica shells are used to filter oils and syrups, thanks to their abrasive qualities. Over billions of years, diatoms have left behind vast deposits of their silica cell walls. This accumulation is what we call diatomaceous earth, which has practical applications in industry. For example, you might have encountered diatoms or golden algae, also known as desmids, which are similar types of microscopic algae. These organisms, especially diatoms, play a crucial role in aquatic ecosystems and have significant industrial uses due to their unique silica shells. Most dinoflagellates have two flagella, which are like tiny tails that help them move. One flagella runs along the length of the cell, so longitudinally, while the other moves transversely or across the cell, often in a furrow between the wall plates. This dual flagella setup gives them a distinctive spinning or whirling movement in the water. Now, these organisms can appear in a variety of colors: yellow, green, brown, blue, or red, depending on the main pigments they contain. These pigments are crucial because they help with photosynthesis, much like plants on land. A really important phenomenon to remember is red tides. This happens when red dinoflagellates, like goniolacs, multiply rapidly. When they do this in large numbers, they turn the sea a striking red color. But it's not just a visual change. These blooms can produce toxins that are harmful not only to marine life like fish, but also to humans who consume affected seafood. Their cell wall is quite sturdy, made of stiff cellulose plates that sit on the outer surface. This provides protection and structural support. Most dinoflagellates are marine, meaning they live in saltwater environments, and they are primarily photosynthetic, so they produce their own food using sunlight. Understanding these features helps us see how dinoflagellates fit into the marine ecosystem, especially considering their role in phenomena like red tides and their significance in the food chain. Euglenoids are fascinating organisms because they can switch their mode of nutrition depending on the environment. When sunlight is available, they perform photosynthesis, just like plants, thanks to their green pigments. But if they find themselves in the dark, they behave more like heterotrophs, preying on smaller organisms to survive. This dual ability makes them quite adaptable. Their pigments are actually identical to those found in higher plants, which explains their photosynthetic capability. Unlike many other microorganisms, they don't have a traditional cell wall. Instead, they have a unique, protein-rich layer called the pellicle. This pellicle gives their body flexibility, allowing them to change shape and move easily. A common example of euglenoids is euglena, which is a freshwater organism often found in stagnant water bodies. They are quite distinctive because they have two flagella, one short and one long, that help them move efficiently through their aquatic environment. So, in summary, euglenoids are versatile, adaptable organisms that combine features of plants and animals, making them a key example of the diversity we see in microscopic life. Let's talk about saprophytic protists, specifically slime molds. Under the right conditions, these organisms come together to form what's called a plasmodium. This is a large, multinucleate mass that can grow and spread over several feet, making it quite remarkable. When conditions become unfavorable, the plasmodium doesn't just die off. Instead, it differentiates, meaning it changes into a different structure. It forms fruiting bodies at its tips, which produce spores. These spores are really special. They have true walls, which make them extremely resistant, allowing them to survive for many years. When the spores are ready to disperse, they are carried away by air currents, helping the slime mold colonize new areas. This whole process highlights how adaptable and resilient these protists are, fitting into the larger picture of their role in ecosystems and their survival strategies. Let's take a closer look at these four major groups of protozoans. First, some of them have a spore-like stage in their life cycle, which is important for survival and reproduction. Moving on, we have the sporozoans, which are interesting because they often lack flagella and are typically parasitic. Next, there are the flagellated protozoans, which are characterized by having one or more flagella that help them move through their environment. Then we have ciliated protozoans, which are covered with tiny hair-like structures called cilia on their surface, aiding in movement and feeding. Lastly, amoeboid protozoans use pseudopodia, those false feet, to capture prey and move around. It's also crucial to remember that all protozoans are heterotrophs, meaning they rely on other organisms for food, either as predators or parasites. These groups are believed to be primitive relatives of animals, giving us insight into early evolutionary steps. Understanding these distinctions helps us grasp the diversity and complexity within the protozoan world and their roles in ecosystems and health. All right, let's take a closer look at this activity. Here, we're asked to match different protozoan organisms with their respective groups. This is a great way to test our understanding of protozoa classification, which is key in microbiology. As we go through these, think about their defining features, like their cell structure, mode of movement, and habitat. Recognizing these characteristics helps us accurately categorize each organism. Remember, understanding these groups not only helps in identification, but also in understanding their roles in health and disease. So, let's focus on connecting each protozoan to its group, based on what we've learned so far, making this exercise both a review and a reinforcement of our overall understanding of protozoan diversity. Let's take a closer look at the diversity within the kingdom fungi. Some fungi live as symbionts, forming mutually beneficial relationships. For example, lichens are a partnership between fungi and algae, while mycorrhizae involve fungi associating with the roots of higher plants. These relationships are crucial for ecological balance and nutrient exchange. Most fungi are saprophytes, meaning they absorb soluble organic matter from dead or decaying substrates. This role makes them vital decomposers in ecosystems, recycling nutrients back into the environment. Now, moving on to their structural features. Unlike yeast, which are unicellular, most fungi are filamentous. Their bodies are made up of long, slender, thread-like structures called hyphae. When these hyphae network together, they form what we call mycelium. This network is essential for the growth and nutrient absorption of fungi. Additionally, the cell walls of fungi are primarily composed of chitin and polysaccharides, which provide strength and rigidity. Finally, some fungi depend on living plants or animals for survival, acting as parasites. They derive nutrients directly from their hosts, which can sometimes cause diseases. So, to sum up, fungi are heterotrophic organisms with diverse lifestyles, structural features, and ecological roles, making them a fascinating and essential part of the biological world. Let's explore how fungi reproduce. Sexual reproduction in fungi involves a series of complex steps: plasmogamy, which is the fusion of cytoplasm, karyogamy, the fusion of nuclei, and then postzygotic meiosis, which results in the formation of haploid sexual spores. These spores are crucial because they allow fungi to disperse and survive in various environments. In groups like ascomycetes and basidiomycetes, there's an interesting stage called the dikaryotic stage, represented as N+N. During this phase, each cell contains two distinct haploid nuclei, one from each parent. This stage is known as the dikaryon, and the entire phase is called dikaryophase. It's a unique feature that distinguishes these fungi and plays an important role in their reproductive cycle. On the other hand, fungi can also reproduce asexually. This process involves the production of asexual spores, which are typically genetically identical to the parent and enable rapid propagation. Vegetative reproduction, which is another form of asexual reproduction, occurs through mechanisms like fragmentation, fission, and budding. These methods allow fungi to grow and spread efficiently, often without the need for spores. All these reproductive strategies, sexual, asexual, and vegetative, highlight the incredible adaptability and diversity of fungi. They enable fungi to thrive in a wide range of environments and play vital roles in ecosystems. Now, let's take a closer look at some examples of smut fungi. As you can see, there are different types categorized into various classes. For instance, Rhizopus is a well-known genus within the Zygomycetes class, often called bread molds because of their common presence on bread. Claviceps is another fascinating example. It belongs to the Ascomycetes class and is notorious for infecting rye and other grains, producing alkaloids that can cause ergotism. Moving on, the rust fungi are a distinct group, classified under the Basidiomycetes, and they are responsible for many plant diseases, especially in crops like wheat and corn. Truffles are also part of the Basidiomycetes class, but they have a unique relationship with symbiotic trees, forming ectomycorrhizae. Then we have yeast, which falls under the sac fungi or Ascomycetes, and they are essential in baking and brewing because of their fermentation abilities. Lastly, the mushroom, which is perhaps the most familiar, also belongs to the Basidiomycetes and is widely appreciated both as food and for its ecological role. Now, as we move forward, I want you to think about how these fungi are grouped into their respective classes. So, for our activity, I'd like you to arrange these fungi into their correct classes based on their characteristics. This will help reinforce your understanding of fungal taxonomy and the diversity within the fungal kingdom. Let's take a closer look at some fascinating plants like Cuscuta, Venus flytrap, and bladderwort. These examples show just how diverse the plant kingdom can be. Now, in plant cells, you'll notice that the cell walls are mainly made of cellulose, which gives them structure and support. One of the most interesting aspects of plant life is their life cycle, which has two distinct phases, the diploid sporophytic phase and the haploid gametophytic phase. These two stages alternate with each other in a process called alternation of generations. It's a key concept because it highlights the complex but organized way plants grow and reproduce. Now, moving on to the broader picture, All these plants belong to the kingdom Plantae, which includes all eukaryotic organisms that contain chlorophyll, enabling them to perform photosynthesis. While most are autotrophic, meaning they make their own food, some have adopted partial heterotrophy. For example, insectivorous plants like the Venus flytrap and bladderwort actually trap and digest insects to supplement their nutrients. Cuscuta, on the other hand, is a parasite, relying on other plants for survival. So, understanding these unique adaptations helps us appreciate the incredible diversity within the plant kingdom and how these plants fit into the larger ecosystem. Now, let's look at heterotrophic eukaryotic organisms. These are organisms whose cells lack cell walls, setting them apart from many other groups. They rely on other organisms, directly or indirectly, for their food, most often depending on plants to provide nutrients. Inside their bodies, they digest their food in an internal cavity, which is a key feature of their digestion process. Once they digest their food, they store energy reserves as glycogen or fat, which helps them maintain energy over time. Their mode of nutrition is called holozoic, meaning they take in solid food particles, digest them internally, and absorb nutrients. An important characteristic of these organisms is their ability to move, what we call locomotion, which helps them search for food, escape predators, or explore their environment. All of these features, being heterotrophic, having no cell walls, digesting internally, storing energy as glycogen or fat, and their ability to move, are fundamental traits of the kingdom Animalia. These traits connect to our overall theme of understanding how different organisms are classified based on their structure and function. Viruses that infect bacteria are usually double-stranded DNA viruses. The protein coat, called the capsid, is made up of small subunits known as capsomers. This capsid serves to protect the nucleic acid inside. When we talk about bacterial viruses, or bacteriophages, they can contain either RNA or DNA as their genetic material. A virus essentially is a nucleoprotein, meaning it has both nucleic acid and protein components, and its genetic material is infectious. Generally speaking, viruses that infect plants tend to have single-stranded RNA, while those that infect animals can have either single or double-stranded RNA or double-stranded DNA. Looking back historically, M.W. Bajerink in 1898 demonstrated that an extract from infected tobacco plants could cause infection in healthy plants. He called this infectious fluid contagium vivum fluidum, which means infectious living fluid. It's important to note that outside of their specific host cells, viruses are inert. They don't do anything until they find the right host. The term virus itself, meaning venom or poisonous fluid, was given by Louis Pasteur. Additionally, in 1892, D.J. Ivanovsky identified certain microbes as the cause of mosaic disease in tobacco, which was a key step in understanding viruses. And one of the most famous examples, the tobacco mosaic virus, or TMV, was among the first viruses to be studied extensively. So, to sum up, viruses are unique infectious agents with specific structural and genetic features, and understanding them helps us grasp how they affect different hosts across the biological spectrum. In 1971, T.O. Diener made a groundbreaking discovery. He identified these tiny infectious agents called viroids. They are even smaller than viruses, which makes them quite fascinating. Unlike viruses, viroids are just free RNA molecules. They don't have that protein coat that typically surrounds viruses. This makes them unique because they rely solely on their RNA to infect and cause disease. The RNA in viroids has a low molecular weight, meaning it's quite small and simple compared to other genetic materials. One of the most important points to remember is that viroids are known to cause specific plant diseases, such as the potato spindle tuber disease, which can have significant agricultural impacts. So, understanding viroids helps us grasp how infectious agents can be surprisingly simple, yet highly effective in causing disease. Now, let's talk about the most common disease caused by prions, which is bovine spongiform encephalopathy, often known as mad cow disease. This disease is particularly concerning because it affects cattle and has serious implications for human health as well. A prion, as you might recall, is a unique type of protein that has the ability to induce normal proteins in the brain to fold in an abnormal way. This misfolding causes brain tissue to develop sponge-like holes, which is why it's called spongiform encephalopathy. Prion diseases are quite insidious because they can impact both humans and animals. In humans, these diseases can lead to devastating neurodegenerative conditions. What's especially alarming is that prions can sometimes be transmitted through infected meat products, which is a key reason why food safety protocols are so critical. So, understanding prions and their role in these diseases highlights the importance of monitoring and controlling these infectious proteins to prevent outbreaks and protect public health. Lichens are fascinating because they are actually symbiotic associations between two different organisms, algae and fungi. The algae part is called the phycobiont, and the fungi part is known as the mycobiont. Now, what's really interesting here is how these two work together. The algae produce food through photosynthesis, and they share this nourishment with the fungi. In return, the fungi provide a protective shelter for the algae and help absorb essential mineral nutrients and water. This close relationship makes lichens very sensitive to environmental changes, especially pollution. They are actually excellent indicators of air quality because they don't grow in polluted areas. Their presence or absence can tell us a lot about the health of an ecosystem. So, understanding lichens not only teaches us about symbiosis, but also about how living organisms can serve as natural monitors of our environment. Let's explore some interesting aspects of plants and microorganisms. Plants are primarily autotrophic, meaning they produce their own food through photosynthesis. However, can you think of any plants that are partially heterotrophic, relying on other sources for nutrients? Some orchids and sundews, for example, have such adaptations. Next, let's talk about the infectious agent discovered by T.O. Diener. He identified the virus called the tobacco mosaic virus. Its key features include a filamentous or rod-shaped structure composed of genetic material, either RNA or DNA, and protein. One notable disease caused by this virus is mosaic disease in tobacco plants, which results in mottled and discolored leaves. Moving on, what happens to saprophytic protists under different conditions? When conditions are favorable, they thrive, reproduce rapidly, and carry out their decomposing activities. But in unfavorable environments, they often form resistant cysts or spores, helping them survive until conditions improve. Now, let's briefly review the four major groups of protozoa. These include flagellates, amoeboids, ciliates, and apicomplexans. Each group has distinctive features. Flagellates move using flagella, amoeboids use pseudopodia, ciliates have hair-like cilia for movement, and apicomplexans are mostly parasitic with complex life cycles. Why are deuteromycetes called fungi imperfecti? Because they lack a known sexual reproductive stage, which is a key criterion for classifying fungi. This makes their classification somewhat provisional until their sexual forms are discovered. Next, a quick differentiation between Plasmodium and Plasmodium. Plasmodium, with a capital P, refers to the genus of protozoan parasites that cause malaria in humans. The term Plasmodium in lowercase can also refer to a mass of cytoplasm in certain organisms, but in this context, we're focusing on the parasite. Let's now look at viruses. They are tiny infectious agents with a simple structure, mainly a protein coat called a capsid enclosing genetic material, which can be DNA or RNA. They are obligate parasites, meaning they need host cells to reproduce. Some common viral diseases in animals include foot-hand-mouth disease, rabies, swine fever, and avian influenza. Turning to fungi, what distinguishes sac fungi from puffballs? Sac fungi, or ascomycetes, produce spores inside sac-like structures called asci. Puffballs, on the other hand, are basidiomycetes that release spores through a pore when mature, often giving a puff of spores into the air. Lastly, both dinoflagellates and euglenoids have two flagella and contain chlorophyll, enabling photosynthesis. However, Dinoflagellates typically have cellulose plates and are mainly marine plankton, while euglenoids often live in freshwater and can switch between autotrophic and heterotrophic modes depending on environmental conditions. That wraps up our review. Each of these points connects to understanding the diversity and complexity of plant and microorganism life, highlighting how they adapt and interact within ecosystems.